Hey, tech enthusiasts! Welcome back to NotSoTechie, where we break down the latest and greatest in technology for everyone to understand. Today, we're diving into a groundbreaking advancement from MIT that's revolutionizing our understanding of the human brain. Imagine being able to see detailed 3D images of entire brain hemispheres. How cool is that? Stick around because you won't want to miss this incredible journey into the human mind. But before we get started, make sure to hit that subscribe button and ring the bell so you don't miss out on any future tech updates. So, the Massachusetts Institute of Technology, MIT, has recently achieved something truly extraordinary: 3 revolutionary advancements that have enabled them to create detailed maps of entire human brain hemispheres. These innovations involve high-resolution imaging of brain tissue at various scales and mapping the connections between neurons with single-cell precision. Are you ready to see how researchers achieve this? Keep watching. For years, neuroscience has dreamed of observing every detail of the human brain while it remains fully functional. This seemingly unattainable dream has now become a reality, thanks to a recent study published in Science. This study unveiled a groundbreaking technology pipeline developed by a brilliant team at MIT. This pipeline allowed the team to process, label, and image entire brain hemispheres of 2 individuals, one with Alzheimer's and one without, with unprecedented accuracy and efficiency. Kwang-Hoon Chung, an associate professor at MIT, studied human brain tissue in detail, taking pictures of individual synapses and entire brain hemispheres. This data is now publicly accessible, opening new doors for brain research. Professor Chung stated that the human brain is extremely complex, with over 100 billion neurons and 200 billion cells, making it one of the most intricate systems in the universe. To understand how the brain works and its issues, it's crucial to understand how the cells are organized in 3D and how they are connected. With each new thought, the brain becomes more intricate. Neurons create numerous distinctive connections with other neurons. It is important to note that every brain is different. Additionally, brains have the ability to change and adapt. Significant progress has been made in trying to access the brain for imaging, but it's more like a cutting process than cracking. Despite the brain's complexity, neuroscience has struggled to penetrate an intact brain. However, a breakthrough was recently achieved by a team from MIT. Professor Kwang-Hoon Chung said that traditionally, there have been 2 main technologies used to study the human brain: advanced imaging technologies like MRI and light microscopy, and molecular labeling methods like immunohistology, which are used for non-invasive in vivo imaging. To map the brain more effectively, Chung and his team of innovators had to improve the technology because both technologies have their own shortcomings. Wow, right? Imagine the possibilities. But how did they do it? Let's explore. Studying the brain to comprehend its activities and impairments. Scientists think that along with billions of cells and neurons, there are probably thousands of different types of neuronal cells in the human brain. Professor Chung said that to understand how the brain works and its issues, it's important to understand how the cells are organized in 3D and how they are connected. Maintaining and retrieving this 3D data necessitates undamaged 3D tissues. Until recently, it was necessary to cut the brain into slices in order to image it at a subcellular level. Chung explained that optical imaging techniques are not capable of capturing the entire human brain due to its large size. Cutting a whole hemisphere of the brain can harm the tissue and take a long time because the slices need to be very thin. That posed an initial obstacle, but also an opportunity to improve and enhance the technology. The entire hemispheres of the human brain have been accurately charted. MIT created a set of data processing tools called a technology pipeline that successfully captured entire hemispheres in 2 donated brains, one of which had Alzheimer's disease. According to Chung, they developed 3 technologies with the potential to fully map the human brain. Although they haven't captured a complete human brain yet, they have successfully imaged different levels of human brain tissue. This includes detailed imaging of individual synapses and entire brain hemispheres. A comprehensive image of the brain, depicting intricate details of neurons, cell structures, and molecules, has never been captured before, from expansive landscapes to diverse forests of cells. Professor Chung highlights the primary challenge he faced while striving to achieve the remarkable progress was building a team of 3 exceptionally talented young scientists at MIT. Every member of this team has played a crucial role in the development of the 3 major innovations, which has rightfully earned them the status of co-lead authors of the paper. Their invaluable contributions have paved the way for significant advancements in the field. MIT is responsible for innovating not just one technology, but three. Let's dig into each one of them and take a closer look. Number 1. Megatomy. Mechanical engineer Ji Wang, who previously worked as a postdoc, created the megatome, a device which allows for the precise slicing of intact human brain hemispheres without causing any harm. The new design of Wang's vibratome slicer ensures that no anatomical information is lost when slicing a brain hemisphere, reducing the time needed from months to complete the process. Number 2. MELAST. After ensuring the slice could survive, the next stage involved the expertise of a skilled chemist. Joo-hyuk Park, a former materials engineer and postdoc, developed a chemical process called MELAST technology. This process makes brain slices transparent, flexible, resilient, and capable of expanding. It also allows for easy and consistent labeling. By improving the efficiency of the slicer megatome, researchers were able to obtain thicker brain slabs. One of the main reasons why thicker slabs can be used in the pipeline is due to the development of MELAST by Park. The hydrogel developed by Park can render brain samples optically clear, virtually indestructible, and able to be compressed and expanded. When used with other chemical engineering technologies from Chung's lab, the samples can be easily and evenly infused with antibody labels that highlight specific cells and proteins. The polymeric system created by Park can modify tissue properties and enable imaging of the entire human brain at different magnification levels. Researchers were able to capture images of an entire hemisphere, right down to individual synapses, in about 100 hours using a specially designed light sheet microscope, according to the study. Number 3. Unslice. After taking images of each slab, they required computational power to convert that data into 3D models. Webster Guan, a software developer and former chemical engineering student at MIT, created Unslice. This system accurately reconstructs both hemispheres in full 3D, aligning blood vessels and neural axons with precision. It creates detailed 3D images of the whole brain by using data from different sections. The images show the restored connections between cells, like blood vessels and axons, with high accuracy. The technology pipeline is truly groundbreaking. It enables neuroscientists to see an entire hemisphere, zoom in on tiny details at the cellular level, and learn more about brain disorders than ever before. The research team showcased the pipeline's abilities to reveal detailed information about neurons, cell structures, and synapses. More images and videos of the study show the versatility of the labeling. They focus on the complex connections between axons and the different types of cells, including neurons, astrocytes, and microglia. They also show the abundance and shapes of these cells. Applications of the new technology. Advancements in neuroscience have allowed for complete imaging of entire hemispheres of the human brain using updated technology. One of the most exciting applications of this technology is in the study of Alzheimer's disease. The team has made progress by comparing brains with and without the disease, finding new details about neuron loss and synapse density. Professor Chung has worked with Alzheimer's researcher Matthew Frosch for years to study and image Alzheimer's disease brains. They recently established a new pipeline for their research, allowing them to explore freely. They found the areas with the most neuron loss in the disease sample compared to the control, and used available technology to investigate further due to their curiosity. This led to a series of detailed investigations. The researchers concentrated their analysis on the orbitofrontal cortex in both hemispheres. They observed that synapse loss was mainly found in regions where there was direct overlap with amyloid plaques. In areas without plaques, the synapse density was high in both the brains with Alzheimer's and those without the disease. Chung explains that with only 2 samples, the team cannot draw any definitive conclusions about Alzheimer's disease. The study shows that it is now possible to fully image and analyze both sides of the human brain. This opens up new possibilities for future research in this field. This new technology platform can be enlarged and will help us understand how human organs work and how diseases develop, leading to new treatments. So there you have it, a revolutionary step forward in neuroscience from the brilliant minds at MIT. What do you think about this incredible advancement? Let us know in the comments below. If you liked the video, please give it a thumbs up, share it with your friends, and don't forget to subscribe to NotSoTechie for more tech updates. Until next time, stay curious and keep exploring the fascinating world of technology. See you soon.